For this lab, we are going to be focusing on bacterial conjugation. This is a 2-week lab traditionally, and so we'll be working through your lab packets, pages 77 up through 83. And then next week we will finish off the lab starting at page 84. So when we're talking about bacterial conjugation, it's important to differentiate between prokaryotes and eukaryotes and how they are able to transfer genetic material. In general, bacteria, which are prokaryotes, they are able to transfer genetic information in a whole host of ways, and so really the variety of methods that they have available to them for exchanging genetic material is much greater than what eukaryotes have. So when we're talking about the transfer of genetic material, when you think about eukaryotes, and one tends to think of transferring genetic material from parent to offspring, that would be considered vertical gene transfer. And so what we're going to be focused on now is horizontal gene transfer, going from one bacterial species to another. With regards to horizontal gene transfer, bacteria are able to do this in one of 3 ways. The first is via transformation. So what's happening here is a bacteria dies and spews out its genetic contents into the surrounding area. And another bacteria can then slightly permeabilize their membrane and take up that the other bacteria's genetic contents, and that oftentimes is either chromosomal DNA or something called plasmid DNA, which are just small circular pieces of DNA. And so that recipient bacteria can either integrate that new genetic material or it can get rid of it. So if it chooses to integrate it or utilize it, it now will have some new genes that it's able to express. So we say that that bacteria has been transformed. The second method is via transduction. This is when a virus infects a bacteria and ends up accidentally incorporating some of the genetic material from the host bacteria, and when that virus goes and infects another bacteria, it actually introduces the bacterial genetic material from the first bacteria into another bacteria. And I'll show you what this all looks like. The third method is known as bacterial conjugation. So this would be kind of the equivalent of bacterial sex. So this is when you have direct physical contact between 2 bacterial cells. One bacteria is going to act as the donor, the other is going to act as the recipient. A proteinaceous sex pilus gets extended from the one bacteria to the other, and genetic material is literally transferred through this proteinaceous bridge. So I will systematically go through each of these processes for you. So the first one, again, is bacterial transformation. So here we have a bacteria that is in the process of dying. It's lysing open, its phospholipid bilayer is breaking open, and all the genetic material is breaking down into small pieces and getting digested and is spewed out into the surrounding environment. Another bacteria in that environment may or may not choose to permeabilize its own membrane and take up those small pieces of DNA. At that point, it can assess and determine whether or not to reject that genetic material or actually go ahead and incorporate it into its own genome. If it chooses to do that, we say that the bacteria is transformed. It now has these new capabilities because it's picked up the gene from another bacteria. So that is bacterial transformation. We actually utilize this all the time in the lab. We do this artificially. We will do something that's not very kind to the bacteria. We will permeabilize their membranes by doing something called heat shock. So we put them in a nice warm water bath and then introduce the DNA and then shock them by putting them into an ice bucket. And that seems to permeabilize the membrane, they will take it up and kind of close up and protect themselves. So it's a very rare occurrence, but we do this all the time in the lab in order to study bacteria. The next method is transduction, and again, this is something that is mediated via a virus. So that's the thing that looks like a spider or a lunar lander, and these are specific viruses that only infect bacteria. So what you can see is that the genetic material of the virus just got injected into the bacteria. Eventually, that bacteria is going to die as a result of the virus's genetic material coming in. So what you see happening now is the virus has forced the bacteria to replicate lots of copies of the genetic material to make the protein coat, and now the bacteria has been lysed open, and new viral particles can now go and infect a new recipient. But what you might notice is that half of the genetic material in the virus, half is purple and half is blue. So what happened is accidentally the virus has transferred bacterial DNA from one bacteria to another, as opposed to increasing just viral genetic material. And I can show you that here on this slide, in which we are able to see that the host bacteria here, it has, if you kind of notice, its genetic material is depicted in purple. And now we have the bacteriophage that is landing on the bacteria, injecting its genetic material into the host. That causes degradation, again, of the host genetic material, and the host will be kind enough to go ahead and replicate lots of copies of the virus's genetic material, depicted here in green. And then it goes ahead and encapsulates that into the newly made viral particles. But accidentally, what sometimes happens is some of that bacterial DNA that had been degraded gets bundled up into a virus's protein coat. So now, once this bacteria is lysed open and all these viral particles are liberated, if you look at this particular viral particle now, if it goes and infects a new bacteria, what it basically is doing is introducing bacterial DNA as opposed to viral DNA. And so at that point, it may or may not get integrated into the host chromosome. But again, it's another way of transferring bacterial DNA from one bacteria to another. However, this method is mediated via a virus. All right, the third method is conjugation. This is the one that I said is basically equivalent to bacterial sex. So we have a donor and we have a recipient. And what is important to point out is the donor has its normal chromosomal DNA depicted here in black. And then it has some plasmid DNA. This is called a fertility plasmid or a conjugative plasmid. And it has the instructions on this plasmid for how to make the sex pilus. So the fertility plasmid, once those genes are expressed on the fertility plasmid, it will go ahead and result in the synthesis of the sex pilus that gets extended to another bacteria, and which is the recipient in this case, and that will then cause the bacteria to come, the two bacteria will come together, and then the fertility plasmid is going to get replicated. And so not just the instructions for how to make the sex pilus, but any other genes that happen to be on that plasmid are now going to be transferred. And so now both bacteria at the end of this conjugation are each going to have the fertility plasmid, and they now will both be able to be donors and transfer the fertility plasmid onto new recipients. Okay, so just to show you how big these plasmids are relative to the chromosomal DNA, here is a bacteria where the genetic content has been spewed out into the surrounding environment. And this whole thing is the big chromosomal DNA. And if you were to spread this out, it'd be in an enormous circle. And now you can see the relative size of a plasmid DNA. So there's a bunch of these small circular pieces of DNA. These are independent of the chromosomal DNA, and they are not necessary for survival of the bacteria, but they can confer survivability under challenging circumstances. So for our experiment today, we indeed are going to be looking at conjugation. And so we are going to have a donor and a recipient. The two bacteria that we're working with today are going to be JM109 and E. coli HY842. So the important thing here is that JM109 has a fertility plasmid. So if we were to draw the bacteria's chromosomes in here, we would draw a huge circular chromosome, and then we would draw a tiny little fertility plasmid, and on that fertility plasmid, in addition to the instructions for how to make this huge sex pilus, it's also going to have instructions on how to confer resistance to ampicillin. So if we were to introduce ampicillin, which is an antibiotic, to this bacteria, it would survive. It knows how to get rid of ampicillin and survive, even though that antibiotic normally can kill bacteria. E. coli HY842, on the other hand, has no plasmids, and so, but what it does have is on its huge chromosome, it has resistance already to some antibiotics, including streptomycin and zeaxanthin. And so we're going to utilize zeaxanthin for our experiments in the next 2 weeks. And so what we are really trying to do is we are going to determine if we can allow JM109 and E. coli HY842, we allow them to incubate together for literally half an hour in an incubator, which is just a warm environment at 37 degrees for half an hour. And we're going to see if JM109 will create the sex pilus and transfer the plasmid that has ampicillin resistance to E. coli HY842. Okay, so I just wanted to point out all the antibiotic resistance genes, so I've kind of depicted that here. So again, we've got E. coli, which is one type of E. coli strain, JM109, and I'll refer to the other one as HY842. And so the JM109, again, has a plasmid, the fertility plasmid, which gives instructions for forming the sex pilus, but it also has the ampicillin resistance gene instructions on here as well. And our recipient in this case is HY842. Now it has no plasmid, but what it does have is it has instructions for zeaxanthin resistance, the resistance to the antibiotic zeaxanthin. And so we're going to allow these two to interact for a short time in an incubator, and then we are going, what we hope is happening is that the donor is copying that fertility plasmid, that it will be transferred, and then we'll end up with a small number of bacteria who have now received the plasmid. So now it's going to be really important for us to figure out how are we going to be able to look at just the bacteria that were transformed, so how can we possibly get rid of the initial donors and the recipients? And so we are going to utilize these antibiotics to kill off all the other bacteria. So if we were to introduce zeaxanthin to our donor, zeaxanthin is going to kill off our donor species. And if we were to introduce ampicillin to our recipient species, they don't have the ampicillin resistance gene, only the JM109 has the ampicillin resistance gene, so we'd kill off this species. Now, if we add both ampicillin and zeaxanthin, the only bacteria that will survive are the ones that initially had that zeaxanthin resistance gene and now have the fertility plasmid that has the ampicillin resistance gene. So in order to select for our trans conjugates, we are going to have to treat with 2 antibiotics, in this case ampicillin and zeaxanthin. All right, so in order to complete the lab for today, I've posted 2 videos on Canvas. One is on aseptic technique, and the other is on the spread plate method. If we were doing this together in lab, this is what we would actually be doing. And then if you could spend some time reading through the lab packet, you need to answer the questions up through page 83. And then on Canvas, I've asked you to actually submit your answers just from page 83, and that is what will be graded. All right, if you have any questions, please post them via email. I'll put a place up on Canvas as well. And I hope you enjoyed this mini lab. All right, take care. Bye-bye.